Photosynthesis is a process which nourishes almost all of the living world. But what is it? Photosynthesis is expressed by the formula 6CO2 plus 6H2O plus light energy yields C6H12O6 plus 6O2, or in other words, carbon dioxide, water, and light energy create glucose and excess oxygen.
Photosynthesis is carried out by C3, C4, and CAN plants. These organisms are known as photoautotrophs. The chloroplast is responsible for carrying out photosynthesis within a eukaryotic cell. Chloroplasts contain chlorophyll, which is the primary pigment used in photosynthesis, and other molecules that function in photosynthetic production of food.
Here is a picture of a chloroplast. Chloroplasts have an outer membrane, an inner membrane, and are composed of tiny little pancake substances called thylakoids. Thylakoids are where the light reactions take place. A group of thylakoids is called a granium. The outsides of chloroplast are called inner membrane space.
One such light reaction occurs in a specialized protein complex known as photosystem II. Photosystem II absorbs photons of wavelength 680. Light energy is captured by the chlorophyll and transferred into high-energy electrons. From here, water is broken up into oxygen, hydrogen ions, and electrons. The electrons are sent to the electron transport chain, and ATP results from the process.
Despite the name, photosystem I occurs after photosystem II. Photosystem I absorbs photons of wavelength 700 nanometers. It's responsible for providing high-energy electrons with which to reduce NADP+, to produce NADPH to be used in the Calvin cycle, which we'll discuss later. The electrons are transferred to a set of clusters where the electrons are then transferred to ferredoxin, a water-soluble mobile electron carrier located in the stroma. This electron transfer creates a positive charge and results in NADP+. When a hydride is transferred to NADP+, NADPH is formed. So the light reactions ATP and NADPH go to the Calvin cycle.
There's actually two different types of chlorophyll, chlorophyll A and chlorophyll B. A is a large molecule that has a porphyrin ring with a magnesium atom at its center. Attached to the porphyrin is a long insoluble carbon-hydrogen chain which interacts with the proteins of the thylakoids and serves to anchor the molecule in the internal membranes of the chloroplast. Adversely, B is an accessory pigment that acts indirectly in photosynthesis by transferring light it absorbs to chlorophyll A.
The next step in photosynthesis, the Calvin cycle, takes place in the stroma of chloroplast. The cycle involves three phases: carbon fixation, reduction, and regeneration. During carbon fixation, each CO2 molecule is incorporated one at a time, attaching themselves to ribulose biphosphate, or RUBP, to form a 6-carbon molecule. This molecule then splits to form 2 molecules of 3-phosphoglyceric acid. Next comes reduction, in which ATP and NADPH are used to convert the 6 molecules of 3-PGA into 6 molecules of glyceraldehyde 3-phosphate. D3P exits the cycle and is sent to the cytoplasm to contribute to the formation of other compounds needed by the plant. This uses 6 molecules of ATP and NADPH. ADP and NADP+ result from reduction, after which the molecules return to the nearby light-dependent reactions to be reused and re-energized. Last is regeneration. Because the G3P from the chloroplast has 3 carbon atoms, it takes 3 turns of the Calvin cycle to fix enough net carbons to export 1 G3P, but each turn results in 2 G3Ps. Thus, 3 turns of the Calvin cycle creates 6 G3Ps. 1 is exported while the remaining 5 stay in the cycle and are used to regenerate RUBP, enabling the system to prepare more CO2 to be fixed. 3 more molecules of ATP are required for the regeneration reactions.